topic today, which is intracellular pathogens, apply common enzymatic strategies to evade host defense system. I must thank my previous and other presenters, particularly Dr. Panda, who made my case and actually gave the history about the AMR and the different kinds of pathogens that have acquired AMR and XDR. So I'm going to talk about some of them, and he also mentioned and provided you with a list of different kind of drug-resistant intracellular as well as extracellular pathogens, which will become, or WHO has actually labeled them as dangerous pathogens for the time being. So I'm going to talk about or touch upon a few things, but before that, let's look at some of the pathogens that we have worked, the intracellular pathogens. Leishmania, which causes Kala-Aza, the tuberculosis pathogen, Mycobacterium tuberculosis, and Neisseria, which causes the very infamous gonorrhea as well as meningitis. So we are going to talk about these intracellular pathogens and the reason behind, because these pathogens, as has already been explained by Dr. Panda, that these are becoming more and more drug resistant and has become a global problem. There are various kinds of multi-drug resistant tuberculosis, multi-drug resistance Leishmania, multi-drug resistance Neisseria. They have been called super gonorrhea, and this is really creating a ripple around the world. So we are all worried about it. So obviously, while people are thinking about looking at it in different aspects, and throughout the day you probably heard about various antibody-related mechanisms through which probably you can target these organisms and take care of it. But then our approach, I being not an immunologist, but a protein scientist, I started looking at it from a different approach. So my approach was looking at what are the antibiotic interacting proteins and why they have become so resistant. So if you look at the global scenario of antibiotic resistance in these organisms, you will see that all those antibiotics targets, that is the molecules to which the antibiotics go and bind, so antibiotic binding proteins, all of them, if you see, have, for example, in penne, pone, RPOB, 23S RNA, and the gyrase, the MTR, which are the efflux pumps, and the porins, PORB, etc. You can see all of them have acquired multiple mutations over time, and that landscape is provided here for you to see. Many of the mutations that are not known have also been worked out here. So one of my students, Shubham, is right here somewhere. So he actually worked this out very recently, and you can actually get a lot of information about the different kinds of mutations that are appearing or have appeared in the antibiotic binding proteins. Now, one of these proteins, however, so one thing is very certain. So the more antibiotic we use, the more indiscriminate use of antibiotics, the more the organism is mutating to avoid their killing. And therefore, we have to look for certain other molecular targets which remains conserved. The more the conserved, the higher will be the possibility of taking care of that organism. So one of those molecules is called asparaginase. We have been working on this particular protein. This asparaginase, I will come back to this slide once again. For the time being, I'm just trying to introduce you about this enzyme. It's asparaginase. It is an amido hydrolase. It converts asparagine. It converts asparagine or glutamine, as you can see here, with the help of this enzyme asparaginase, to aspartic acid and ammonia. Now, aspartic acid and ammonia both are very important intracellular products, which will be utilized later on. I will show you in some other slides. One of the uses of this enzyme is that this enzyme is anti-leukemic in property, and it is marketed by various companies. So anti-leukemic, it works as an anti-leukemic because it kills or it deprives the cancerous cells, the blood cancerous cells, which is leukemia, in a very specific way, because it deprives the cancerous cells which are proliferating from a very important amino acid, which is the asparagine, because it works on the asparagine and depletes asparagine, and therefore those cells do not survive. However, this is present in most of the intracellular pathogens. Some of the pathogens, like Leishmania donovani, the ones that I named, Helicobacter pylori, Mycobacterium tuberculosis, Salmonella typhi, and Neisseria gonorrhea. In all these organisms, this particular enzyme is present, which was quite kind of surprising. And when we looked at the global scenario of how this enzyme is behaving over evolution in terms of its changing or mutations in particularly the drug-resistant varieties, we found that it is absolutely conserved. What it indicates is that this particular enzyme is of special importance for the survival of this pathogen. And we realized that very soon when we looked at the molecular biology of these pathogens. So if you look at Leishmania donovani, you can see that this pathogen survives, initiates as a promastigote, goes and enters into the macrophages, and then finally divides inside the macrophages in an acidic environment and comes out and starts reinfecting. So it goes through a cycle of promastigotes to amastigotes, and from the amastigotes it again keeps on infecting other cells. Now, what happens in Leishmania, if you look at there is one of the very important or common drug against Leishmania is amphotericin B, which is shown here. Amphotericin B represented by AMP-B. Now what happens is as soon as the AMP-B is or amphotericin B drug is injected to a Leishmania infection, the Leishmania actually takes it up, the amphotericin B, through the aqueous pores. Now, along with the aqueous pores, the amphotericin B is taken inside, but at the same time, some of the protons and calciums are also taken inside from the extracellular environment. That causes an alteration of the pH inside the host, inside the pathogen, and that altered pH is required for the phagosome and lysosome fusion, and that will clear up finally the organism. But then these organisms are also very, very smart. What they do is they produce this particular enzyme called L-asparaginase, which we call LDA1, Leishmania donovani asparaginase 1, and that in turn we know converts asparagine to aspartic acid and ammonia, and it is the ammonia that then neutralizes the acidic pH, the H+ ions, and the aspartate and glutamate are utilized for its conversion to other proteins, and therefore this entire pH mediated onslaught is immediately taken care of by the organism. So this is how we realize that Leishmania can actually survive inside the phagosome. If we proceed further, if you look at Mycobacterium tuberculosis, here also what happens is it goes from the phagosome, and ideally the phagosome should fuse with the lysosome, and that requires a particular lowering of pH, and that's a very common method that happens inside the macrophages. But then if you look at this particular section where the phagosome is shown and the mycobacterium is inside, and if we zoom into it, what happens is the phagosome with the mycobacterium inside, as it happens in the case of Leishmania, it also invokes the possibility of many protons or the ATPase to be activated, channeling in a lot of protons inside this phagosome, and the mycobacterium, therefore, through its secretory system called T7 secretory system, they again produces this enzyme called MTA, or Mycobacterium tuberculosis asparaginase, and this asparaginase converts asparagine to aspartic acid and ammonia. The ammonia again neutralizes the situation, and therefore the acidic environment which is necessary for the phagosome and lysosome fusion is again deactivated. So Mycobacterium also uses similar kind of mechanism. Now, if you look at, so these are the two organisms which actually infect and goes and resides inside the macrophages. Now there are organisms which resides or goes and first infects the neutrophils. One of those organisms is the Neisseria. Now what happens when a pathogen enters into a neutrophil? This happens via the pathogen recognition receptors. Immediately it will release a lot of interleukins. These interleukins causes some pathogen recognition, the polymorphonuclear neutrophils to come together, more and more of them, and they will finally opsonize the pathogen, and then they will be killed. So if you look at the phagosome, and imagine that those dots which are shown on the other side, the yellow dots, those are the pathogens, those enter the phagosome, and then as it happens in other cases, here also as soon as it enters, as soon as the pathogen enters the phagosome, it invokes the possibility of generating a lot of H plus inside the cell, lowering the pH. These H plus in turn, in case of neutrophil, actually activates the NADPH oxidase. This NADPH oxidase utilizes oxygen and produces reactive oxygen species. The reactive oxygen species in turn produces hydrogen peroxide or hypochloric acid and myeloperoxidases. All of these at the same time actually have various kinds of azurophilic granules. So this process actually activates the azurophilic granules and they releases these kinds of toxins or antimicrobial peptides and enzymes. Altogether, that is the HOCl, the hydrogen peroxide, MPO, all of them along with the antimicrobial peptides now attack the pathogen that has entered into the neutrophil and clears it off in a process by apoptosis. However, in case of Neisseria, which are XDR, MDR or otherwise also, these Neisseria, when they enter into the neutrophils, they releases the interleukins. The interleukins causes PMNL efflux. This efflux induces release of more interleukins, and then ideally it should be opsonized and cleared off. But then, Neisseria, just like any other organism, are very smart. They have certain proteins, such as the ABC transporters, the pili protein, the LOS, and other proteins, so lipooligosaccharides and other things, and these, and the purines, and these have a special tendency to interact with the, or inhibit the functioning of the NADPH oxidase. So even if there is high influx of protons through these proton pumps are unable to actually activate the NADPH oxidase, and if this doesn't happen, activate the NADPH oxidase in the presence of these purines or some other proteins that are present on the surface of the pathogen, and therefore this entire mechanism is blocked. It doesn't produce the hydrogen peroxide, it doesn't produce the oxidative environment, and at the same time, because it doesn't produce the oxidative environment, then azurophilic granules also cannot interact, and therefore what happens is there is accumulation of protons inside the cell. So although the other processes, which is NADPH oxidase mediated oxidative environment and the azurophilic granules are stopped, but then there is still the cell's possibility of survival because the environment becomes very acidic where the pathogen cannot survive. But then that is where the pathogen again comes into play and produces this enzyme called produces the enzyme called L-asparaginase, and L-asparaginase converts L-aspartic acid, asparagine to aspartic acid in ammonia. The ammonia neutralizes the environment, and again, the pathogen survives. So that is what you can see. I have made the case to actually show you that L-asparaginase is a very common and conserved enzyme in most of the intracellular pathogens, which helps the pathogen to survive inside the cell's acidic environment. So coming back, L-asparaginase, which is used for different things, and then there were some reports that suggested that Mycobacterium tuberculosis exploits asparagine to assimilate nitrogen and resist acid stress. Taking cues from there, we looked at the asparaginase present in all the organisms. We found that they are present in all the intracellular organisms that we were working on. So we thought of cloning and purifying and characterizing this protein. So this is Leishmania donovani protein, this is Mycobacterium tuberculosis protein, and this is the Neisseria gonorrhea protein. So you purified the protein. It's a 42 kilodalton protein, as you can see in all the cases, and its activity against glutamine, against asparagine, its pH, its activity in terms of Km, its efficiency, etc. Everything has been calculated. In the interest of time, I'm not going to detail of that. What essentially we saw by doing all these biochemical assays is that the L-asparaginase show a peak at 70 kilodalton, which is a dimer, in all the three cases, which is Mycobacterium tuberculosis, asparaginase MTA, Neisseria gonorrhea asparaginase NGA, and Leishmania donovani asparaginase LDA. Now all of them show substrate specificity to L-asparagine, but in case of Neisseria gonorrhoeae, they showed a specificity to L-glutamine as well. The optimum pH for activity of all these enzymes was 8. We looked at, because I am a protein chemist, we looked at the global shape of this enzyme, this very important and conserved enzyme through Sachs, and this you can see the structure which shows that it is a dimer. I'm not going to details of this one, but we looked at the sequence of this enzyme and found that this enzyme works as a dimer. There are 2 active sites. The active sites were relatively conserved, and as you can see, it works like a canonical hydrolase, which consists of an acid, base, and a nucleophile. So, and in all the organisms in the lower half, as you can see, all of them are conserved. The sequences of the active sites are absolutely conserved, and they were falling perfectly structurally on top of each other. This is the mechanism how the enzyme works, but then what we do, we now know that L-asparaginase is an important enzyme. How does it work biochemically? We know the structures. Now what to do? Obviously, the next question is to find inhibitors of this enzyme. So we started looking for inhibitors and using computational biology, which again was emphasized. So we do a lot of computational biology and structural biology in our lab. So we did a primary screening on the zinc library and also did on the traditional Chinese medicine library. We did a secondary screening, toxicity testing, and finally screened out certain molecules for each one of these enzymes. The molecules, in the case of Leishmania donovani, we called them, oh, sorry, there is a change over here. So Leishmania donovani, we called them L1, L5, L1, L2, L5, L7, and L12. And for the Mycobacterium tuberculosis, we called them M3, M26, M15, 29, and 31. And in the case of Neisseria gonorrhea, however, is what is interesting observation. We found that certain molecules which are binding to Neisseria gonorrhea L-asparaginase were actually already FDA-approved drugs, and they were the best binders. And to them, those were like the pimerolast, thalidomide, and decitabine. And these we named N2, N8, and N9 for brevity. The binding constants of each one was decided by various means. Using also molecular dynamic simulations, we found that these drugs are binding and capable of inhibiting the activity of L-asparaginase. These are all data. If you want to go into details, I can actually tell you more and more details, but in the interest of time, I'm not going. So, the first one, I will just explain the first one here. What we are plotting on the x-axis is the hydrogen bonding capacity, and on the y-axis, we are looking at the salt bridges. So in case of LDA1 alone, that means Leishmania donovani asparaginase alone, these salt bridges and the hydrogen bonding capacities of the 2 active sites are quite far apart, and you can see them as 2 peaks. But if you look at when L1 or the inhibitor of asparaginase was used, then these 2 peaks converge, essentially suggesting that the inhibitor is working very effectively. And in case of LDA2 also, or L2 also, it is also working. In case of Mycobacterium tuberculosis, we looked at the RMSD, root mean square deviations, with respect to the holoenzyme or the only enzyme, and with respect to present of these inhibitors. And you can see there was sufficient inhibition. In case of Neisseria gonorrhea also, we looked at various inhibition propensities, and these are data which can be obtained from simulation studies. Now, when you report such things in any of the journals, the journals obviously come up with another question. So they are always like, moles, the field moles, chuhe, pahadi chuhe jo hote na. So these moles are like that. They always keep on asking questions. So if you block certain questions, it will come out from another hole somewhere else. So it's always like that. They are jumping around and they keep on finding some or the other loophole. So here you find, you ask, you do something, you present it, the paper comes back asking this question, you shut them off, then another question, and you do that, you keep doing that. That's how research progresses. So we keep on doing that, and that's how we came to look at the enzyme inhibition and binding kinetics. So this was like, you have done fantastic work on just in silico biology, why don't you do some biochemistry? So we had to do this biochemistry, and here we show that, the Leishmania donovani can be L-asparaginase can be inhibited effectively by L1 and L2. In the case of Mycobacterium tuberculosis, they can be inhibited by the M26 and M29. In the case of Neisseria gonorrhea, this can be inhibited by all the 3 N2, N6, and N9 molecules that we found out through simulations. So we looked at not only the simulation, we looked at the binding by ITC. isothermal titration calorimetry, and there also we found interesting data, and with very high binding capabilities. So these are, yeah, yeah. So in the case of, oh, okay. Yeah. So in the case of Neisseria gonorrhea also we found that these are binding with very high capability, and we looked at the binding studies we did with for the Mycobacterium tuberculosis through ITC, and that was also giving good binding data. And yeah. Somehow, so the data related to cell studies where the actual Mycobacterium tuberculosis inside the macrophages, the Neisseria gonorrhea, and the Leishmania donovani cultures were grown, and then that was given the drugs one after the other in a dose-dependent manner. You could find inhibition. That slide is simply not showing because of some technical. I can see it here, but not there. There's not some problem. And these are because I have accumulated or put all the data together for the different studies of Leishmania, the Mycobacterium, and the Neisseria. So these are the people who worked. These are the papers we published, and we started in 2015, and we are continuing on that study. And thank you very much for your patience.